Hello everyone, welcome to yours and my favorite part of the week. That's right, it is your online general psychology lecture, and I'm here to walk you through the hottest topics in biopsychology. Now, this chapter is a little listy. I do have a bunch of applications of kind of explaining how specific brain regions are related to specific disorders and just kind of like common drugs. But a lot of this is kind of like, okay, we kind of need the basic understanding of the brain, which is a little dry, but this is kind of our last chapter that's kind of dry and broad. That foundational knowledge that you kind of need to understand how the brain works and how the brain is related to specific behaviors, which will be useful in later chapters. Okay, so with that said, let's jump into it. Biological psychology or biopsych, it doesn't matter, it's more or less the same thing. Just different names for the same thing. It's a great thing about psychology. Often happens. So neuroscientists and biological psychologists have made huge strides in understanding how the brain works. Researchers who study the relationship between the nervous system and behavior. These scientists help bridge the gap between the nervous system and our behavior, allows us to span multiple levels of analysis. So we talked about levels of analysis in previous lectures, and again, I have the same figure here, and kind of conceptualizing this as an elevator, wherein each of these being a floor, and you're looking outside on an elevator, and as you go up each floor, and imagine like a see-through elevator, and you're looking downward at the outside world through those glass paneled elevator. And you can basically what this metaphor is alluding to is that obviously the outside world isn't changing, but as you move up and down the elevator, you're kind of getting a bit different perspectives on that same outside world. And it's kind of the same thing here. So for biology, what allows psychologists to do is really focus on how hormones, neurotransmitters, kind of the brain kind of more generally, impacts is associated with certain behaviors. I should also state that it's going on at kind of a genetic level as well. So all kind of that stress research that everybody talks about, a hot topic being like cortisol, this is kind of what that level is looking at, kind of measuring those hormones and seeing how they're related to real-world circumstances. Cells of the nervous system. Learning how the body's cells and organs function can help us understand the biological basis of human psychology. The nervous system is composed of two basic cell types: glial cells, also known as glia, typically called glial cells, and neurons. So kind of the point here is that behaviors just don't occur magically. They're related to specific neurological activity in certain brain systems and really also different kind of genetic systems, which we're really not going to go into depth about in this chapter, but kind of the same principle here. So really understanding the biological basis of human behavior. So a key question: what the hell is a neuron? Well, neurons are brain cells that specialize in communication. They can be characterized as communication portals. They are the central building block of the nervous system. Neurons communicate electrically and chemically. There are around 100 billion neurons in your brain and about 160 trillion connections between them. A number that's not even, you can't even imagine that number. It's beyond human capacity to understand what 160 trillion means. But that's how complex neuroscience is. It's trying to, and it kind of speaks to how difficult and how little, frankly, we know about human behavior. And this is just one example here, trying to investigate 160 trillion connections. And not just those raw connections, but they work in sync. They have these very complex developmental networks of brain regions, of neural activity that is associated with behavior. So trying to unpack that is a very difficult task. And these numbers kind of speak to that. Parts of a neuron. So let's do a very basic understanding of what's parts of a neuron here. We're going this is kind of like old school, like 7th grade biology, but here we're going for neurons. So beginning with the cell body, known as the soma. It is the center of the neuron. It builds new and renews cell components. It's where the nucleus of the neuron is located. That's really the key part here, right in that cell body. Dendrites are branch-like extensions that receive electric information from other neurons. They're kind of like receivers on your phone. They listen into other neurons and pass on information to the cell. And you can see that kind of in the left corner, or leftist corner, dendrites. They're kind of like branches, as stated. And as stated, they listen to what other neurons are saying, and that's kind of how they receive information from other neurons through the dendrites. The axons, or the tails of the neurons, that spread out from the cell body and transmit electric information, can be several feet long. They're phone transmitters. They send messages to other neurons. So, kind of, again, broadly speaking here, this is just a very basic understanding of neurons. But we have the dendrites, which are kind of like receivers on the phone. We're receiving the call. They're listening to other information, and then they're kind of passing it on. And axons, meanwhile, are our cell phone transmitters. They're sending information to other neurons. And axons are kind of to the right, you can see on this figure, of the soma, the cell body, and they go all the way down until they eventually reach the terminal buttons of the neuron. And what are the terminal buttons? Well, it's a knob at the end of the axon that contains synaptic vesicles filled with transmitters. And what are synaptic vesicles? They hold neurotransmitters. You can think of synaptic vesicles as gel capsules holding medicine, so like a general pill that you take. So they have kind of like, well, kind of straightforward here, like a gel like container that holds neurotransmitters. And what are neurotransmitters? They are chemical messengers that allow neuron-to-neuron communication. So kind of our neurons are doing two functions. They're receiving information from other neurons through their dendrites. And you can kind of see that in the upper left-hand corner. And then they're transmitting information through the axon to the terminal button. The terminal button is kind of where a bunch of synaptic vesicles are, again, which hold neurotransmitters, and neurotransmitters allow for chemical communication between neurons. Because remember, we have electrical communication and we have chemical communication. This brings us to the synaptic cleft. It is a small space between neurons where communication between neurons occurs. The neurotransmitters bind to receptor sites on the dendrites of the receiving neuron. So we have this, really what the cleft is kind of that open area. If you can imagine, and going back, you can look at the previous figures of neurons next to each other. There's kind of a gap between different neurons. And what happens in that gap is the neurotransmitters are kind of dumped into that little gap. And what the neurotransmitters do then is kind of attach or bind to a neighboring neuron. So one neuron releases neurotransmitters, and then those neurotransmitters enter that little space, and then they attach to a different neuron. And that's one way that they communicate those neurons through a chemical process, again, neurotransmitters. Now, what are receptor sites? Proteins on the receiving neuron surface, the neurotransmitters attach. They vary in shape, with different shapes matching different neurotransmitters. Really, the only thing you need to know about receptor sites is that they allow neurons to communicate with each other. So these on the bottom of that figure is the dendrite of a receiving neuron. So again, one neuron sending out a chemical message in the form of neurotransmitters into that little vacuum space, known as the synaptic cleft. Where the neurons get kind of dumped off, then the where the neurotransmitters get dumped off, and then the neurotransmitters attach to the other neuron receptor site located in the dendrites. And how those neurotransmitters attach to that other neuron is kind of like a lock and key system. You can imagine that each receptor is kind of like a keyhole, and each neurotransmitter is a key. And so only specific neurotransmitters can fit within specific receptor sites. The neurotransmitter and the receptor have what is referred to as a lock and key relationship, as I stated. Specific neurotransmitters fit specific receptors. Similar to how a key fits a lock, the neurotransmitter binds to any receptor that it fits. And there's a much more detailed breakdown of this in the textbook. This is kind of the broad overview, and these kind of large bullet points of the general function of these different parts of the neuron is what I'm looking for. Just kind of a general understanding of, okay, how do these neurons communicate chemically, and what are some of the parts of this, or what are some of the parts that are a part of this chemical communication process? And glial cells will go into far less detail. They can kind of be conceptualized as support cells, kind of really supporting helping out the neurons. They play a role in the formation of myelin, which we will talk about. They remove debris, kind of clean up waste, and they enhance learning and memory. There's many glial cells as neurons in the brain. So going back to all those billions of neurons, well, there's an equal amount of glial cells. Now, these actually used to be kind of a side note. This is not quizzed on, but these things used to be thought of as kind of like an afterthought. They're really not doing anything. But now there's a better understanding of them because, again, we know very little about the brain. And it's suspected that actually these glial cells play a very important role in kind of the functioning of the brain. But don't worry about that for this quiz. Just a little background information. The myelin sheath, we already referred to the glial cells kind of playing a role in helping create this. And in the other figures shown, you can see the myelin sheath kind of on the axon of a neuron. So what is it? Well, it helps insulate the axon. It increases speech of neurocommunication. It's like hopscotch. Neural signals jump from node to node. And the node is those little small gaps. You can see them here in between the sheets. So they're kind of like, you can imagine them kind of like a blanket around the neuron. But the blanket does not cover the entire axon. Again, that long tail coming out of the cell body. But rather, there's little gaps in between those blankets, those myelinated sheets. And how information is, or how neurons communicate electrically is kind of like, as stated, like hopscotch. So what does, and we'll break it down more momentarily, is basically electrical signals jump from one of those nodes, those little gaps, to another. And that really speeds up the process because, again, we have trillions of neural connections and you're constantly moving, you're talking, you're thinking. So these things are firing constantly. And by firing, I mean like just being active constantly. So it has to be kind of very fast. Now, interestingly, multiple sclerosis, an autoimmune disorder, involves a large-scale loss of myelin sheath on axons throughout the nervous system. The resulting interference in the electrical signal prevents the quick transmission of information by neurons and can lead to a number of symptoms, such as dizziness, fatigue, loss of motor control, and sexual dysfunction. It's kind of like a phone signal becoming hopelessly scrambled. There's no known cure. So because of the damage to those myelinated sheets, kind of the communication that the neuron is trying to do with its neighboring neurons electrically is disrupted. And it's kind of like trying to talk into a phone, and you're talking to someone and maybe they're under a tunnel or they have very bad reception and it's going in and out. It's a similar concept here. The other neighboring neurons can't really figure out what that neuron is saying electrically. And thus kind of get confused, and it can lead to various symptoms: dizziness, fatigue, loss of motor control, etc. And you can see on the left-hand side a normal nerve, being referred to as kind of a neuron. And on the right side, you can see a neuron with multiple sclerosis. And you can kind of see, it kind of looks like there's a bite in the myelin in the sheath, which is an indicator of multiple sclerosis. So that's kind of like the biological basis of, or one part of the biological basis of multiple sclerosis. That deterioration of, that damage to the myelin sheath, creating communication problems between neurons, which is associated with various symptoms seen in multiple sclerosis. All right, so now let's jump more specifically into electric communication. So we already talked about chemical communication with those neurotransmitters, kind of entering that vacuum space, being released by one neuron, I should say, then entering that little vacuum, that synaptic cleft, and then attaching itself to those neurotransmitters, then attaching itself to another neighboring neuron, kind of allowing which signals or I should say allows for chemical communication between neurons, because, and we'll talk about momentarily, different neurotransmitters are associated with kind of different behaviors and different kind of biological processes. But now, jumping to electric communication, neurons respond to neurotransmitters by generating electrical activities. We know this is true because scientists have recorded electrical activities from neurons using electrodes. Electrodes allowed researchers to measure the electrical charge inside versus outside the neuron. So once a neurotransmitter is released into that vacuum, and then attaches to the receptor cells of a neighboring neuron, that generates an electrical signal in the neuron who received that neurotransmitter. And we know this is true because we can attach electrodes to a person's scalp and kind of track electrical changes that can be measured by literally putting electrodes on your scalp. And what is basically going on is when a neurotransmitter attaches to that receptor site of that receiving neuron, this starts a biological process wherein all these kind of different ions, these molecules, enter that receiving neurons, or I should say enters that receiving neuron, and that kind of, these different charged ions then change the electrical makeup of the receiving neuron. So basically how this works is that in general, when there's no kind of neurotransmitters being received by a neuron, it's all neurons are kind of operating at negative 55 volts. So it's all, and in that state, when a neuron is at negative 55 volts, it's basically doing nothing. It's just sitting there, and nothing's really going down. But when that receiving neuron receives a neurotransmitter, then, as stated, all these different molecules enter that neuron, and that changes that voltage within that neurotransmitter from negative 55 volts to a more positive charge, so higher than 55 volts. So basically, it hits zero, eventually hits like 40 volts. So basically, that's the process. A receiving neuron receives a neurotransmitter. That neurotransmitter then kind of allows these charged ions to enter that neuron, which then changes the voltage of the neuron, which then starts kind of this electrical process wherein communication occurs between neurons. And bringing back up that figure on that we had in the previous slides, and then expanding on this a little bit, when a neuron receives signals at its dendrite, again, the dendrite is kind of like your cell phone receiver, you're receiving information, that's where the neighboring neurotransmitters are attaching to. The receiving neuron's gates open, allowing positive sodium ions to enter. Neurons receive signals when a neurotransmitter from a neighboring neuron binds to a receiving neuron receptors. Ions are charged atoms. So this is basically what I've already stated. The neurotransmitter binds to the dendrites of a neighboring neuron, which then allows these different positive ions, or these different ions with different charges to enter that neuron. The neuron's electrical charge changes, causing the receiving neuron's electrical voltage to increase from negative 55 volts to about approximately, eventually 40 volts. So from going negative volts to positive volts. And then eventually, when there's enough of a charge inside the neuron, so once it hits a certain number of voltage, an electrical impulse called an action potential is triggered. It is an electrical impulse that travels down the axon, triggering the release of neurotransmitters. Abrupt waves of electric discharge triggered by a change in the charge inside the axon. This is the universal language of neurons in humans and all other animals. They're how neurons communicate. This is the neuron firing. That is what it means when people say neuron is firing. It's referring to this electrical communication in neurons. It's an all or nothing response. It's kind of like a gun. You either shoot the gun or you're not shooting the gun. So it has to hit a certain voltage level for the neuron to fire, and then send, which when I say fire, it means that they release an electrical impulse from kind of the cell body in that dendrite area all the way back down to the axon. And then that axon then connects eventually, that electrical signal, I should say, eventually reaches the terminal buttons of that neuron, and then releases neurotransmitters, which then connect to another neuron, and that's basically the process. Neurons can fire, though. What I'm explaining, though, is that is happening thousands of times per second. This is going on for billions of neurons. In between firings is a very brief absolute refractory period. The neurons cannot fire again until after this period. So in the top right figure, you can see that the resting potential, which is kind of, as I stated before, is when the neuron's really not doing anything. But then all of a sudden, the neuron, the receiving neuron, receives that neurotransmitter. Then all of a sudden, those ions are entering the neuron. The voltage is changing. It's going from that negative 55 to negative 60 range voltage. And then that neuron is increasing in voltage. That electrical charge is becoming more positive. And you can see in where number 2 is on that figure, in the right-hand corner, that the voltage is increasing, and boom, action potential happens. That then releases electrical charge down the axon, eventually causing neurotransmitters to be released. But you can see also in that figure number 3. So after that action potential happens and the electrical impulse is released or generated from kind of that cell body range and down the axon, the voltage then will go way back all the way down to its previous resting potential, that negative 55, negative 60 range. But you can actually see that it overshoots it. It gets too negative. It goes below that negative 55, 60 range to almost like negative 80. And what that actually means in practice is that now the neuron can't do anything. It cannot fire again. It cannot release another electrical impulse until its voltage increases a little bit again. So it actually overshoots itself. So it's at resting potential. It receives that neurotransmitter signal. Its voltage starts to go up. Action potential happens. It releases an electrical impulse. Then the neuron's voltage then starts to decline, going back to that 55 rate. But it says, oh shit, I overshot the voltage. Now I'm at like negative 80. I now can't do anything, so I have to take 5 minutes to recover and get my voltage back up to negative 55 before I can fire again. And that's basically the process. So you can really see how kind of the chemical part, the neurotransmitter part, and the electrical part kind of work together to allow your neurons to communicate at a very, very fast rate, which allows for very complex human behavior. Now I want to talk a little bit about reuptake, because it's related to a very real-world application. But before we get there, so inside neurons, communication is electrical. As we demonstrated, that entire electrical action potential is occurring within one neuron. But between neurons, it's typically is chemical via neurotransmitters. So we already talked about that. Chemicals being released, or neurotransmitters being released into the synaptic cleft, and that's how the neurons are communicating with each other. But within the individual neuron, the action potential, that electrical signal, eventually culminating in the release of neurotransmitters. So within a cell, it's electrical communication, but between cells, chemical communication via neurotransmitters. Again, this is very broad, not exactly true, but good enough for this class. Now, an interesting example that I want to talk about is this process called reuptake. So this process of communication between cells is halted by reuptake. Reuptake involves the neurotransmitter being pumped back into the neuron that released it in order to clear that synaptic cleft area. And really what this is doing is kind of regulating the production of the neurotransmitter. So basically what that means is that say a whole bunch of, we'll just make up a number here. Again, this is completely made up. Say 5 dopamine neurotransmitters are released into the synaptic cleft, but oh wait, there's only 4 receptors available in the receiving neuron for dopamine. So there's 5 dopamine neurotransmitters, only 4 receptor dopamine areas available, receptor sites available in the receiving neuron. That means there's 1 extra dopamine neurotransmitter. So what the neuron will do, who actually released the dopamine, it'll basically just suck it back up like a vacuum. It'll take back that dopamine neurotransmitter, and that way it doesn't have to, generate and create a new dopamine neurotransmitter. It's kind of like a way of recycling. The neighboring neuron can't use this dopamine I produce. Let me just suck it up like a vacuum, and then I don't have to create a new dopamine neurotransmitter, which requires energy and resources that your body doesn't want to waste. Now, interestingly, where this becomes more applicable in the real world is SSRIs. That is, for example, selective serotonin reuptake inhibitors. This is a medication. Typically, you can see this done. Most common example, I should say, is with people taking these types of drugs when they have depression, but there's various other mental health disorders associated with SSRIs. So kind of it's basically kind of limit this process by which, as stated, neurotransmitters entering that synaptic cleft, that kind of vacuum area wherein they attach to a neighboring neurons, receptor sites. And then if no receptor sites are available, the neuron who actually sent out that neurotransmitter, in my example, dopamine, kind of recycles it back up. What SSRIs do is kind of limit that recycle process. They limit reuptake, or they inhibit reuptake, meaning they limit it. So what it does basically is the SSRI drugs, they kind of force the neurotransmitters thought to be associated with kind of depression, such as serotonin, to stay in that synaptic cleft area. Or, which allows them, those neurotransmitters, more time to find kind of like a receptor site to attach to, thus kind of increasing the amount overall of like serotonin or any neurotransmitters kind of circulating through the body. So basically that's what kind of SSRIs are doing. They're trying to limit the reuptake and increase the amount of specific neurotransmitters, in the case for depression, serotonin, in that synaptic cleft, thus, maybe stabilizing a person's mood, decreasing kind of their negative mood, for example, by increasing the amount of serotonin within that synaptic cleft, by decreasing, by inhibiting reuptake. And that is how SSRIs work, not exactly, but close enough. Now moving on to other types of medications and how they relate to neurotransmitters. Scientists have developed specific medications to target the production or inhibition, that is to decrease the amount of neurotransmitters. Opioids bind with opioid receptors and mimic endorphins to reduce pain. Xanax reduce anxiety by stimulating certain neurotransmitters called GABA receptor sites. You don't need to know that. Driving down neural activity. Prozac inhibit the reuptake of neurotransmitters, which we covered, serotonin, allowing neurotransmitters to stay in that synapse or that synaptic cleft area longer than usual. So what medical professionals, researchers can do is, again, kind of design medications that target specific neurotransmitters. They can increase the amount of a neurotransmitter, or they can decrease the amount of neurotransmitter within an individual to try and help them with various neurological or psychological disorders. And specifically kind of looking at the opiates, for example, there are opioid receptor cells within humans. And what opioids did or does is kind of mimic and kind of copy our natural forming endorphins, which exist within our body. So we naturally produce these endorphins, which allow us to reduce pain. Well, what the drug opioids do is kind of bind that lock and key of that drug, of that neurotransmitter that scientists are creating. Those opioids actually fit within the receptor sites of those endorphins, thus allowing kind of the production. It's a kind of a synthetic way, a scientifically manufactured way to kind of make or increase the amount of endorphins within a body, within someone's body, thus reducing pain. So that's how opioids target pain, is by kind of mimicking the properties, having a similar lock, so they can fit within the key of those endorphin, those opioid receptors. And you don't need to know about the GABA receptor sites. And we already kind of talked about Prozac again, kind of with depression. That's by inhibiting reuptake of serotonin, allowing serotonin kind of be in that synaptic cleft area longer. And those are kind of the two kind of big examples of how drugs mimic kind of naturally forming or created neurotransmitters within the brain, or not just mimic them, but also kind of stimulate the production of, or limit the reuptake in the case of Prozac. Now I want to talk about actually what is serotonin. And serotonin influences arousal. Specifically, it deals with mood and temperature regulation, aggression, and sleep cycles, which, all really kind of related to, depression and various other mental health disorders. Thus, those drugs are kind of designed to target that neurotransmitter. Another common neurotransmitter, dopamine, which plays a role in our response to rewarding experiences and aids in motor function. Now, I wanted to say briefly here that one interesting and challenging part of designing drugs that actually work is that serotonin, dopamine, and all these other neurotransmitters that I'm not going to talk about, they do multiple things. So they are involved in all kinds of different cycles. And that's where you get these side effects coming in, because you can target serotonin, for example, but to try and improve or stabilize mood. But at the same time, serotonin isn't just involved with stabilizing mood. It's involved with all kinds of other functions. Thus, when you take that drug, you have all these other, you can have other side effects, because that you're affecting neurotransmitters, not just, or specifically serotonin, not just the parts of the serotonin that deal with mood, but just serotonin in general. You're targeting serotonin, and as stated, serotonin is in charge of or involved in all kinds of different functions. Thus, you get various side effects with drugs. So that's kind of the real world applicability of why it's so challenging to produce effective drugs, just because these neurotransmitters do so many different things, and as stated, there are trillions of neuron connections. It's very difficult to just be like, okay, I want to target this one neuron. I don't even know if it's possible. So that's kind of some of the challenges of designing medicines that target neurotransmitters and kind of treating depression, for example, medically, and through drugs specifically. Next, I want to briefly talk about neuropeptides. Neuropeptides are kind of like neurotransmitters, but you know how I was just saying that neurotransmitters are involved with all kinds of different things? Well, neuropeptides are kind of more focused, more specialized jobs. And I'm also specifically bringing it up because I want to talk about one neuropeptide only, and that is anandamides, or I should say anandamide. So nailed that pronunciation. Anyway, anandamides are cells in our bodies, including neurons, manufacture anandamide, which binds to the same receptors as THC. They influence eating, motivation, memory, and sleep. THC is binding to the naturally produced anandamide receptor sites. This could be one reason behind Why? When people smoke marijuana, medically, of course, in the state of Florida, it's associated with munchies and being sleepy. So kind of the idea here is that, so basically what THC does, is kind of an active ingredient within marijuana, is it attaches to kind of these naturally existing and across numerous animals, actually, not just humans, anandamide receptors. So the THC attaches to these anandamide receptors, and what do those anandamide receptors or what are those anandamide receptors associated with? Well, when those receptors are activated, the activation of that receptor is associated with eating, memory, motivation, and sleep. So kind of the idea here is that THC attaches to those kind of naturally forming anandamide receptors within the brain, and thus you, a hypothetical you, is likely to experience munchies and being sleepy, falling, taking medical marijuana, of course. So that is kind of the biological process of THC. THC attaching to those naturally forming neurological receptors, increasing the likelihood of being hungry and being sleepy. Next, I want to talk about something called neuroplasticity. Plasticity can be defined as nervous system's ability to change slash adapt over time. So it's called plasticity because you can imagine it as plastic. So like you can form plastic like a hot plastic basically, and you can manipulate it to any shape that you want it to be. Thus, over time, neuroplasticity, therefore, refers to the fact that the nervous system, the brain, more generally, can adapt, can change, can be shaped over time by environmental influences. So some scientists speak of brain circuits being, quote, hardwired when they don't change much, if at all. Despite what the popular media often implies, very few human behaviors are, quote, truly hardwired. Claims about language, jealousy, morality, or other human capacities being hardwires should be treated skeptically. We're genetically predisposed towards these abilities, but they aren't fixed, let alone predetermined. The nervous system is continuously changing by leaps and bounds, as in early development, or more subtly, as with learning. So basically what this is getting at is just the realization that through time, as we develop, go from a fetus to an older adult, neurological changes are undergoing. And those are kind of a naturally occurring process, but also due to learning and just experiences in our real world, our environments, impacting our brain development. It's changing our brain. It's and thus kind of the framing, the wording of something being quote unquote hardwired is generally not accurate. Most things, most psychological psychological outcomes are influenced in one way or another by the environment. Again, reflecting the plasticity of the brain. You can imagine, for example, that humans, when they're born, obviously, they're incapable of basically doing nothing. But compared to something like a giraffe or a goat or something, like day one, those animals, they're running around, they're doing something. While humans take forever to develop. And that's got to be because of this plasticity process, wherein the environment is impacting the development of our brain. It allows for your brain to kind of be specialized in based on the environment that it's living in. And those are kind of just conceptual points. You don't have to memorize that or anything, but that's basically what we're getting at here, the ability of the brain to change and adapt over time. Now let's talk about some examples of neuroplasticity. During learning, our brain changes. So literally listening to this lecture is hopefully allowing some learning opportunities, wherein your brain changes. And some specific ways that learning changes. Generating new synapses generating increased connections and communication among neurons. So what I mean by synapses is, as stated, connections and communications among neurons. So those, remember when I talked about those connections, those whatever it was, 16, those trillions of connections? Well, during learning, new connections are formed, which allows for kind of increased communication, increased efficiency, and allows you to do more complex thinking and more complex tasks. Also, something called long-term potentiation occurs. That is, it strengthens already existing synaptic connections. It makes neurotransmitters release into the synaptic cleft, produce a stronger and more prolonged response. So kind of we have two processes here. We're generating new connections, new synapses, but we're also strengthening the connections that already occurred. There's something also called neurogenesis, which is the creation of new neurons, which occurs during adulthood. It used to be thought that, oh, you're only born with X amount of neurons, and those are what you have for basically your entire life. But now scientists argue, the consensus is that you actually can produce new neurons across the lifespan potentially. And I'm going to get, and what's marked in red is stuff that I'm not going to test on. But kind of the idea here, though, is that there's a limitation on neuroplasticity, a limitation on how well the brain can adapt. So, for example, if you have a very serious brain injury or serious illness, the brain is not able to adapt to allow it to maintain its normal functioning. So if you have a serious injury, the brain tries to adapt, but if it's super serious, you lose a big chunk of your brain, the brain will struggle to adapt sufficiently enough for it to maintain prior levels of functioning, prior to the serious injury or illness. So there's a limitation on the neuroplasticity. And I have here 4 ways in which neurons change during development, but I just want to focus on 2. The first being pruning. This consists of the death of neurons. They remove connections that aren't useful. So we already talked about the formation of new connections, new neurons, long-term potentiation that is strengthening already pre-existing connections. However, the brain also has this naturally occurring process where death of neurons occur and connections are actually removed if the brain determines them not to be useful. During pruning, as many as 70% of all neurons die off. This process is helpful because it streamlines neuron organization, enhancing communication among brain structures. So kind of the idea here, and the general thought about brain is like, or the brain is like, the more activity, the better, and it's kind of like the pop psych way of thinking about things. But really, that's not the case. The brain is really structured to be highly specialized and be as efficient and kind of communicative as possible. So what this pruning process allows is that it kind of removes synaptic neural connections that aren't really serving a beneficial purpose. So it kind of reduces the amount of cooks in the kitchen, for example, which allows more of kind of a streamlined process and allows for greater communication, allowing you to perform more complex tasks due to kind of this increased efficiency. This is also kind of related to myelination, which we already talked about, which increases the kind of the speed of a neural connection. So we have the speed of neuron connections through myelination, and then we have increased efficiency by the brain pruning off irrelevant, unnecessary connections, but at the same time identifying connections that are useful to you. And strengthening those and forming more connections in parts of the brain that are related to tasks that you perform routinely. So for example, say I'm a mathematician and there's a part of my brain, I'm just making up something that does math, and all I do is math all day. You would expect to see a ton of new and strong neural connections in math and kind of a pruning in other areas kind of not associated with math. Again, this is very broad, not exactly accurate, but that's what this is getting at. I want to bring in a couple examples. One, rodent examples. Rats exposed to enriched environments such as large cages with multiple animals, toys, and running wheels develop more elaborate dendrites with more branches than do rats exposed to a standard environment of a cage with only 2 animals and no objects. So you can see on the left side and the right side the 2 different neurons, and you can see how many more branches, how many more dendrites the enriched condition produces in the rat. So basically what this is getting at, again, going back to the environmental point, is that your environment impacts your neurological development. In this case here, you have two sets of rats. One rat's just kind of living in kind of a shitty cage alone, or maybe with one other animal. Not a lot of stimulation, not a lot of things that rats like doing, versus living in like Rat Disney World, and you're having the time of your life. You have an enriched environment wherein they're kind of engaging all their cognitive processes. They're playing on the wheel. They're playing with other rats, having a great time, and that kind of enrichment can be seen at a neurological level. In this case here, with increased dendrites, which allow for increased communication, again, increased efficiency, and increased kind of ability to perform complex tasks. So this enriched environment allowing for greater, a higher number of dendrites in the rat. And then an example, in humans, certain brain regions can sometimes take over the functions previously performed by other brain regions, just as some players of a sports team can often take over for an injured player. For example, in blind people, the capacity to read Braille, that is a system of raised dots that correspond to letters in the alphabet with their fingers, is taken over by brain regions associated with vision in sighted people. So kind of the idea here is that a person becomes blind and then learns Braille, so now reads through running their finger across dots on a page. And what's interesting is that when a blind person is reading Braille, the same brain regions associated with vision in non-blind people and sighted people are active in that blind person. So literally the vision parts of the brain, broadly speaking, being activated when a blind person is reading Braille. So literally that brain function, that a brain region associated with vision, helping to compensate, kind of taking on a new role for that person. Kind of like taking over from an injured player on a sports team, which is part of kind of the neurological basis by which blind people can quote unquote read. Literally, the brain regions associated with vision being activated while reading, quote unquote, seeing Braille. So an example of, so that's an example of neuroplasticity. That brain region recognizing, oh, I'm blind, on a biological level, of course, saying, oh, I'm blind, therefore I'm going to adapt, I'm going to take on a new role, and my new role is for vision. I will apply my vision features, my specialization of that brain region to reading Braille. Next, let's talk about specific brain regions. Sensory information comes into and decisions come out of the central nervous system. Think of our nervous system as a super highway with a two-way flow of traffic. And we'll break that down more briefly in the upcoming slides. Let's begin with something called the forebrain, though. The forebrain is the most developed area of the human brain. It's responsible for analyzing sensory processing and higher brain functions. It's involved with planning, language, and complex decision-making, things that you think of yourself as human. It's 40% of the volume of your brain, which is a gigantic portion. It contains 12 to 20 billion neurons. So that's kind of how complex and how critical this brain region is, the forebrain, and how makes kind of you human, because this part of the brain taking up so much, being 40% of your total brain area, the forebrain being 40% of your total volume of your brain, can represent again how complex this is and how important it is to the brain, because it requires a lot of energy. It takes up a lot of space. When something biological is taking up a lot of space, requires a lot of energy, that means it's evolutionarily very important. So the forebrain consists of two cerebral hemispheres connected by the corpus callosum, which allows communication between them. They're kind of like two figure skaters in pairs competition. They communicate and cooperate continually. So really all you need to know here is that this forebrain, a big important area, is involved with these higher order, it's called higher order processing, that is like planning, language, those complex things that make us human, those kind of things. And then this corpus callosum, which is kind of like this highway across two hemispheres of your brain. You have two hemispheres in your brain. Sometimes old school pop psych would say, oh, I'm a left hemisphere thinker, or I'm a right hemisphere thinker. And it's really not, that's really not true. That's kind of a myth. Really, the entire brain works together in kind of a complex way. And even really these specializations I'm going to go over are not exactly correct. Like they have different brain regions do have some specialization, but for any activity, numerous parts of the brain are activated, and they're kind of working in unison. So it's really not, okay, I'm doing this one task and this part of my brain is working right now. That's not really accurate. The brain works in kind of these dynamic systems, allowing for complex behaviors to occur. And in the case of the forebrain, the corpus callosum is basically allowing the basically two parts of the forebrain and located in different hemispheres, the left and the right, to kind of communicate. It's like this big highway of nerves that allows for kind of rapid communication between these two parts of the forebrain. In the forebrain, the largest part of the forebrain, I should say, is the cerebral cortex, again, involved with planning, language, complex decision making, kind of the things that make us human. It can be divided into 4 lobes. Each is associated with a different function. So let's walk through the 4 lobes of the cerebral cortex. And the cerebral cortex is kind of talked about a lot in general media, the exposure of psychology. So it's kind of useful to know some of these lobes. Beginning with the frontal lobes, it oversees and organizes most other brain functions. This is referred to as executive functioning. So kind of like how the US president exerts control over the members of his or her cabinet, the brain's executive function provides a kind of top-level governance over cognitive functions. It oversees other parts of your brain. Moving on to the motor cortex part of the frontal lobe. So within the frontal lobe, there's something called the motor cortex. It's responsible for movement, so motor movement, literally. Each part of the motor cortex controls a specific part of movement. More precise movements require more space in the brain. The prefrontal cortex, which is another part of the frontal lobe, is responsible for thinking, planning, and language. You can find Broca's area here. It's a language area that helps to control speech production. So really all you need to know here is that the frontal lobe kind of has this executive kind of role, overseeing other parts of your brain. You can really think of this as like your consciousness kind of thing, wherein you're overriding maybe moods that you have, you're making yourself focus. It's kind of like that deliberate thought process. It's an overseeing process. And within that frontal lobe, the motor cortex is responsible for motor movements. And the more complex the motor movement, as stated, the more space it requires in the brain. So if I'm doing like a double backflip, I'm doing a 720 backflip, that requires more space in the brain than, just moving my finger, because it's a complex task. And then we have the prefrontal cortex. And really, the only thing you need to know there is, again, it's kind of related to this consciousness, thinking, planning. And really, for the purposes of this class, you don't need to know the Broca's area. Just focus on kind of the broad overview of the frontal lobe. And I want to bring up a case study. This is a classic case study. It's not that great, but I mean, it's taught, so I'm going to talk about it in this class. Phineas Gage. Basically, Gage was a railroad foreman who experienced a horrific accident in 1848. His job was to build railroad tracks through rural Vermont. Gage was performing his usual daily work task, filling holes with gunpowder to break up stubborn rock formations. He was pressing gunpowder into one hole with a tampering iron when an explosion suddenly propelled the iron with great velocity through his head. The iron pierced Gage's face, which you can see on the right-hand side, under his cheekbone and destroyed his prefrontal cortex. Later, computer analysis suggested that the rod largely passed through or entirely through his frontal lobes, and specifically his left frontal lobe. Physician analysts at the time in 1848, this is Old English, Gage was indulging at times in the grossest profanity, which was not previously his custom. His mind was radically changed, so decidedly that his friends and acquaintances said he was, quote, no longer Gage. So kind of the idea here, this is supposed to be a classic case study of wherein you have this nice guy who's just some railroad worker living his life. People like him in general. All of a sudden he takes an injury. He takes a metal rod through the prefrontal cortex. Because of that, he becomes impulsive. He's cursing. He can't control anything. He's not really planning. So kind of like those responsibilities, the functions of the prefrontal cortex are no longer functioning. The prefrontal cortex is no longer functioning, and therefore he's going to have different behaviors because the brain regions responsible for not cursing at your boss, or doing whatever, just being completely irresponsible, are no longer functional. Thus, you're going to have, because those brain regions don't work, thus you're going to have behavioral consequences. So this is kind of the case study, an example of what happens when you lose your prefrontal cortex. However, it's generally accepted now that Gage's personality changes may not have been as drastic or as long-lasting as sometimes assumed. For years following the accident, Gage worked as a successful stagecoach driver in Chile, and a doctor who examined him around this time found him to be largely normal. This is a reminder of the brain's plasticity and its often astounding ability to recover at least some of the function, even in the face of devastating damage. Gage's case occurred in the midst of the 19th century debate over localization, regarding whether certain functions of the brain are associated with particular functions. So basically, this is why I don't like teaching a lot of seminal studies, because they end up kind of not being true ever. So now there's growing evidence that Gage really didn't have that dramatic of a personality change. Of course, we didn't have fMRI and all these brain imaging technology we have, now 200 years ago. So this could be kind of a case of maybe he did have those initial profanity latent kind of impulsive behaviors, but over time maybe the brain adapted, again representing the neuroplasticity. So maybe different parts of the brain compensated for damage to the prefrontal cortex to make him more or less impulsive over time. But also, the scientists at the time could have also been over-exaggerating their claims, because there was a big debate at the time of how does the brain work? Does it work through these very local, each part of the brain does this part of the brain does this part of the brain does Y, whatever, etc. So it's called localization, a very specialization view of the brain versus this network approach wherein the brain is, all parts of the brain are working together to do complex tasks. So maybe that scientist was a localization person, so he's like, oh no, he lost his prefrontal cortex, therefore he lost his planning ability, his thinking ability. See, look, I'm right. I'm a localization scholar. This is evidence that I'm correct. So maybe whoever the physician was at the time may be biased due to this debate. And kind of shows an example of how your historical period, the ongoing debates, can kind of influence your interpretation of results. So the results are results, but your interpretation can change based on various biases, which everybody has really, but we've developed since then kind of ways to help kind of control for those biases. So we talked about the frontal lobe. Now let's talk about the parietal lobe. And specifically, I just want to talk about its specializations for touch and perception. Really, touch and perception is what I want to focus on here. It helps track objects' locations in space. It relays information to the motor cortex every time we reach, grasp, or move our eyes. Again, the motor cortex being in the frontal lobe. So again, kind of demonstrating how these different brain regions work together. Now, interestingly, damage to the parietal lobe can result in something called unilateral neglect. Neglect opposite side of the body from where the damage occurs. So say you're a right parietal lobe. We both have left and right parietal lobes. For all these lobes, we have a left and right. Say your right parietal lobe is damaged, you can experience unilateral neglect to the left side. So it's always opposite. So if you're damaged on the right side, you experience neglect on the left side. If you have damage to your left parietal lobe, you experience neglect to the right side. Now, what is neglect? Neglect is it means that an individual in human, or not just human, but other animals, such as a dog, which you'll see in this video, can no longer perceive objects in space on a certain part of their vision. So say again, I have damage to my right parietal lobe. I no longer can perceive kind of things to my left-hand side. So what you'll see here for people with this who have unilateral neglect is if you put a bowl of food in front of them and say, again, I have damage to my right parietal lobe, therefore I can't perceive things on my left side. What you'll do is if you put a plate of food in front of those people, they'll only eat food on their right-hand side because they have neglect on their left-hand side. So they literally, like, they're not perceiving the food on the entirety of their food. They're only perceiving food on the right-hand side. If they have damage to the right lobe. So what this can actually lead to is old people who have, or anybody really, who has this damage of people starving to death because you'll feed them food and they'll become undernourished because they'll be like, oh, I finished my plate of food, and really they're only eating half of their food because they cannot perceive the entirety of their plate. And again, look at the video for a more kind of vivid illustration of what's going on. Now let's move on to the temporal lobe, the lower part of the cerebral cortex that plays a role in hearing, understanding language, and storing autobiographical memories. The temporal lobe contains the auditory cortex and the Wernicke's area, which is responsible for speech comprehension. Severe damage to Wernicke's area results in difficulties understanding speech. People who have damage to Wernicke's area often speak in gibberish because a person doesn't understand the words coming out of their mouth. So for example, if a researcher was, or I should say, when asked what the saying, strike while the iron is hot means, a person with damage to their Wernicke's area or their temporal lobe responded, and quoted, quote, ambition is very, very end determined. Better to be good into post office, in piler box, into distribution, into mail, in survey, in headmaster. So literally, they have damage to this temporal lobe, their Wernicke's area. It causes a person not to understand the words coming out of their mouth. So they think that they're saying the correct words, but what's coming out of their mouth, because they can't comprehend verbal words, is just gibberish. So again, ask, hey, what does strike while the iron is hot means? You ask a person with Wernicke's damage, and they just start spitting word salad because they cannot understand language. They have a lower ability to comprehend speech than people without that brain damage. So really all you need to take away here is that damage to the temporal lobe causes or is associated with difficulty comprehending speech and speaking gibberish because they don't understand the words coming out of their mouth. And finally, the occipital lobe lays at the rear of your brain and are specialized for vision. So if you bang your head on the back of your head, it's stated that you're seeing stars, and that's probably because the occipital lobe is involved with vision, and it becomes activated if you smash yourself in the back of the head. Thus, you're literally seeing something because this vision part of your brain is activated because you got punched in the back of the head, and thus you see stars. Humans are visual primates, so this takes up a lot of space. So as I stated before, the more important a behavior is, or a sense is, in this case vision being very important in humans, the more space it's going to take up in your brain because it's evolutionarily valuable. All right, so let's move to a whole different part of the brain. Let's get out of the cerebral cortex. Let's talk about the limbic system. The limbic system processes information about our internal states, such as blood pressure, heart rate, respiration, perspiration, as well as our emotions. So you have kind of those basic biological processes, things that you're really not thinking about. You're not controlling your blood pressure, for example. How much you're sweating in general. You're not controlling those things. But it also is associated with emotions. So the limbic system is a set of highly interconnected brain regions. It is thought to be the emotional center of the brain. Role in smell, motivation, and memory. Odors invoke memories of emotional memories. Sense of smell projects directly to the limbic system. PTSD can be triggered when exposed to similar odors. So kind of the idea here is that the limbic system evolved in smell, motivation, memory. And why that's interesting is that it kind of helps explain why odors evoke specific emotional memories. Because smell, the sense of smell kind of communicates directly with the limbic system. So which kind of allows for positive and negative memories to be associated with smell. So kind of memory being wrapped up in your sense of smell. And interestingly, you see this with people who have PTSD, wherein they can be triggered when exposed to similar odors of where their PTSD, their traumatic event occurred. So that's kind of the biological basis of why smell can elicit memories. I'm going to skip over thalamus, but we're going to look at hypothalamus, though. It regulates and controls internal bodily states. It controls the pituitary gland. So basically what this is hypothalamus is involved with is really the regulation of these really basic human traits. Sexual drive, thirst, fear, aggression, very, very kind of basic feelings and reactions, really. And kind of the joke is that the hypothalamus regulates the 4 Fs, with the 4 Fs being feeding, fighting, fleeing, and the amygdala, kind of a very common known brain region, just thought to play a role in fear, excitement, arousal, kind of generally thought to be associated, which is kind of emotion in general. Kind of so a specific part of the limbic system thought to be invoked with just emotion. Not really tested on that for the purpose of this class, but I want to talk about hippocampus. Plays its role in spatial memory, so kind of like your memorization of maps and kind of where you are in space. Damage causes inability to form new memories, and it may explain why a portion of the hippocampus is larger in London taxi drivers than in non-taxi drivers, and especially large in experienced taxi drivers. So some research has found that looking at the hippocampus of taxi drivers and then comparing it, their hippocampus and the activity of their hippocampus and the size of their hippocampus to non-taxi drivers, the researchers found that taxi drivers have larger hippocampuses, which kind of illustrates the role of spatial memory. Why would it do that? Because you're a taxi driver. You have to know where the streets are. You have to have kind of a mental map of the makeup of the city. So it would be expected to, kind of going back to neuroplasticity, that brain regions that you use a lot would have be possibly larger in an individual, in this case hippocampus, but kind of in general though, having kind of more and stronger connections of neurons within that brain region. Again, representing how your environment, the things that you learn, the things that you practice impact your structure of your brain. Moving on to the endocrine system. The endocrine system is a system of glands and hormones that controls secretion of blood-borne chemical messengers. Hormones are chemicals that are released into the bloodstream to influence organs and glands. They differ from neurotransmitters because they are transmitted by blood rather than by nerves, that is neurons, which are slower. So blood transportation, that is hormones, are slower than neurotransmitters. It just takes longer time kind of to circulate through your blood. So the endocrine system actually is talked about a lot in kind of the general public. So let's walk through some specific hormones and glands that are commonly talked about. Beginning with the pituitary gland, which controls the other glands in the body, kind of characterized at times as the master gland, and kind of very common to have pituitary gland issues. It releases hormones that influence growth, blood pressure. It includes oxytocin, which is responsible for a number of reproductive functions involved in maternal and romantic love. Now, oxytocin is kind of characterized as like this magical love drug in kind of pop psych, but that's really not the case. To the extent that it does much of anything, or that extent that it kind of boosts sensitivity to social cues, both good and bad. So if you're already feeling bad, oxytocin can kind of like amplify that. But if you're already feeling good, oxytocin can also amplify your sensitivity to good social cues around you. And kind of the idea that you can there's all these like oxytocin like things that you can like sniff and stuff. Not really true. It doesn't really do anything. I know like one of the like leading researchers in oxytocin. A lot of the research on it is, or the stuff that's kind of sold is largely just garbage for not regulated at all. So don't waste your money in general on that kind of stuff. The adrenal gland, which release adrenaline and cortisol during states of emotional arousal. Cortisol typically characterized as a stress hormone in stress literature, but it also is involved with other things. Again, this is not tested on, but it's involved in like growth and other things as well. It's not just the negative bad drug. And in fact, stress often is good. You need some stress to motivate you to learn. Dropping off a kid at kindergarten for the first day of school, yeah, you're a little stressed, but guess what? You're going to get over it. Adrenaline boosts energy production in muscle cells, but restricts it in other cells. So adrenaline in general, you have an adrenaline rush, and then you're kind of full of energy. And muscle cells is kind of the general thought for this. Moving on to sex hormones, testes in males and ovaries in females. Both males and females manufacture testosterone, male sex organ, and estrogen, female sex hormone. All right, now all that bio stuff is done, let's talk about some conceptual ways of mapping the brain, understanding different parts of the brain. One of the earliest ways was phrenology, which was one of the earliest, but was discredited by the mid-1800s, mainly due to early neuropsychology studies of persons with brain damage. Phrenology argued that bumps on the skull corresponded to brain enlargements. Those brain enlargements were linked directly to psychological capacities. So on the right-hand side, what they basically believed was, I can put my hand over your skull and just rub your skull, and then say, oh, there's some bumps on your skull here. Those bumps mean that parts of your brain there are very large. Therefore, you're very good at, you're very hopeful. You're very benevolent. You're very cautious, whatever. So it's kind of just like this feeling your skull and then coming to conclusions based on about your neurological makeup based on your skull. And obviously, through history, this was also associated in practice with like a lot of racism, a lot of anti-Semitism, because you would look at someone's head and you'd be like, okay, that person who is Jewish or that person who is black has a messed up, has a different skull than a white skull. So therefore, I can tell that that person has a different neurological makeup, making them, in general, less human than me. So, but obviously, this was been discredited. Now I'm going to move on to some modern ways of mapping the brain. And each way has strengths and weaknesses. And a lot of studies actually use different parts, or all kinds of different techniques, because they understand that different ones have strengths and weaknesses. Looking at EEG, measures electrical activity via electrodes placed on the skull. It's non-invasive. It can measure quick, rapid changes in brain electricity, kind of those action potentials, in milliseconds. Also can tell someone is awake or asleep. We'll talk about why in later chapters. As EEGs have some disadvantages, so they show kind of an average neural activities that reach the surface of the skull. They tell us little about or anything about what's happening inside the neuron. So you're not, and because it's non-invasive and it just sensors on your skull, you're not looking inside the neurons. You're just reading kind of the electricity that can be picked up through the skull, which is substantial, but it's not really telling you what's happening inside of an individual neuron. So it's not good at telling you where exactly in the brain electrical activity is occurring, but really it's really good at telling you when something's happening, because it picks up electrical changes at a millisecond level. And on the right-hand side, you can see some examples of EEGs. And then next I want to talk about PET scans, which measures consumption of glucose-like molecules to give a picture of neural activity. When neural activity goes up, it increases the intake of glucose, sugar. So neural activity requires sugar. What researchers do is injection of radioactive glucose-like molecules. Scanners measure where in the brain these glucose-like molecules are being used. So you basically inject somebody with these kind of glucose-like molecules that light up in the scanner, so you can like literally see them. And then you can look at the PET scan images and then determine where or which regions of the brain are active during a specific task. So I inject you with some of this radioactive glucose, which is fine, you'll be all right. And then which then reaches the brain, and then when your brain is taking, or parts of your brain are increasing activity, they're using more glucose, and thus these glucose-like molecules that are like multicolored will also promulgate and gather in the brain region that's active, and then the PET images allow you to look at those multicolored radioactive glucose-like molecules in a photograph. So you can see this in the lower right-hand corner. So the brain becoming active, eating that glucose, and then that being visual within the PET images. And next, fMRI, which uses magnetic fields to visualize brain activities. So when brain cells brain cells need more oxygen when they increase activity, so not just glucose, but they need more oxygen. And what fMRI does, it measures changes in blood oxygen levels, indirect indicator of neural activity. Very detailed information, but if you move in the scanner, the data is bad. So basically what the fMRI machine is doing is measuring change in the levels of blood oxygen in a certain part of the brain, which is kind of an indirect indicator of neurological activity. So so that's kind of how fMRI works. You're not doing like this direct, okay, one-to-one, your measuring brain activity, but rather kind of the promulgation, the increase of blood oxygen levels in particular parts of the brain. And basically what you can do is that you can administer a task to an individual and then say, okay, this part, the motor cortex lights up, or the motor cortex is active when running, therefore the motor cortex is involved with motor movement, specifically running, as evident by the elevated levels of blood oxygen within the scanner. But in the scanner, that running example would never work because in the fMRI machine that you're put in, you basically can't move. If you move, the data becomes kind of messed up and you have to basically throw it out. And our last technique, transcranial magnetic stimulation, TMS, applies strong and quickly changing magnetic fields to the surface of the skull. It can either enhance or interrupt brain function. It's good at telling us what areas are involved in different psychological processes. For example, if TMS temporarily interrupts the temporal lobe and the subject displays language impairment, we can tentatively conclude that this lobe plays a role in language. It's an opportunity to infer causation. So what this machine can do is literally enhance activity or interrupt activity in kind of certain brain regions, and then have individuals perform certain tasks. And if they perform better or worse on particular tasks, you can say, oh, that brain region is associated with whatever. So for example, going back to, remember that Wernicke's example in the temporal lobe wherein that person's speaking gibberish? Well, let's say a participant comes to the lab, they don't have any damage to their temporal lobe or the Wernicke's area, but then they go on this TMS machine and say the researcher sets the TMS machine to interrupt your temporal lobe, and then all of a sudden the participant who had no temporal lobe damage starts speaking gibberish. Well, then you can tentatively conclude, oh wait, the temporal lobe is playing some role in language, and which allows you for some opportunity to infer causation about brain regions. All right, everyone, that's all for this week. Thank you so much for listening. I hope that was semi-bearable, and I'll see you next week. Thanks so much. Bye.